to my presentation. Okay, got it. I have to say got it. Okay. So, welcome everyone today, and sorry for a bit of delay because running through the meeting rooms, but I thought to give a presentation on fair play, mastering metadata to empower early career life scientists. And that is how you can enhance your research impact through effective data management. I'll give very simple practical tips. So first, I would like to, yeah. So what is, just to give a small introduction, like I think must know, everyone now should be more aware of FAIR, but let's look at FAIR in context of data. So again, there are two types of data, which is non-qualitative and qualitative data. What is non-qualitative data? That means like you just have an observation, which is recorded as is. For example, if a person has a height, so you record the height as 1.78 centimeter. But what is qualitative data? So qualitative mostly is observation, which is qualitatively analyzed in respect to something else, right? For example, are you taller than me, or someone is taller than me? So that is more like qualitative analysis. Then qualitative data can be recorded as a person, as a group of people, a historical event, a natural event, a document, artifact, or books, for example. So it is non-field specific, but mostly it's an introduction, which is more like related to any field, for example. But more importantly, it's important to collect these things as digital objects. Why? We will come to it later. And that means you have to make it more fair. Now let's split into two types for research data management convenience. So one is human related qualitative data collection and other is non-human related, which is sensitive data and non-sensitive data. Again, these things are a bit, so things change when it is sensitive data and non-sensitive data. So let's keep that in mind and we go further. Now there is in this data processing, how do you process your data? And then how do you later store your data? So data processing can means like analysis, cleaning of the data and things like that. So for that, we need good security, we need infrastructure and things like that. Okay, so now we are in the field of life sciences mostly. I think mostly who have joined are life scientists, but if someone else, I'm sorry, this is more life science data types. So this is basically what are biological data types. So I have tried to cover as many as possible, like genomic data, taxonomic data, environmental data, proteomics data, metabolomics, but data types are just not limited to these. It can also be protocols, your lab methods, the statistical models, the data management plans, educational materials, your presentation you go and give somewhere. So these all come under data, just not the data that you produce in experiment or you make like a code or something. Now, with so much being said, there is an explosion of data in life sciences, right? We know how much data is being produced and everyone is talking about, should we have a place to store data? If someone comes to us, it's always like, where can I put my data in? Do you have a long-term storage, for example? So, and the amount of data is growing exponentially with new machines coming in. Now you can do genomic sequence very fast. You have like terabytes of data being produced a lot, right? So I lost my screen. Okay. Can you see my screen?
Yes, we can see your screen.
I have lost it somehow. Okay. Oh, I don't know. I have lost my screen here. I'll try to go back to it. Okay. Maybe it's just best not to give. Okay, I'll take this off and just do it on my laptop. Okay, now I am back. Okie dokie. Sorry for this. Okay. So, right. So then there are these FAIR guiding principles. So if you see in the FAIR guiding principles, this is a bit of, it goes into a bit of detail, but if you see like every different principle states metadata with respect to data, right? So metadata are assigned, for example, in Findable, globally unique persistent identifiers. Data is described in rich metadata, but I don't want to go in so much detail. You can go through it later when you get the slides. But what I would say is that metadata should be made machine actionable as possible. That means, but PDF or DOI does not mean fair. We have to keep that in mind, right? Now, keeping that in mind, I will just give a brief gist. So findable means like through in context of metadata. I'm not discussing about other data types, but mostly metadata. So findable is like through metadata and persistent identifiers, accessible, for example, via protocols for humans as well as machines, interoperable by using different vocabularies, ontologies, and semantic technologies, reusable through clear licensing, provenance, and contextual metadata. So basically what we focus on is FAIR makes digital objects machine readable, actionable, fostering better integration, discovery, and collaboration research. Now, GoFair gave these FAIR guiding principles, but there are various tools available. So you can have like FAIR, Aware, Fuji for checking the fairness of your data as well as metadata, right? So to be useful for others, data should be fair both for both machines as well as humans. Now, keeping that in mind, let's go here. So why are we so much talking about FAIR principles? Why are we so much concerned about these? Because all the policies, people who are defining the policies these days are really looking at FAIR. For example, UNESCO has given recommendation on open science. EU has open science policies. European Research Council has one. Then let's come to Sweden. Sweden have passed research bills, which are very much talking about open research data implementation by 2026. Government has given assignment to VR, right? Then there are also university policies and things like that, right? Okay, then let's go to VR. So VR has given certain guiding principles for open access, but let's not focus on everything, but just the points that are important. For example, metadata should also be published with open access. So you see the importance of metadata here. The publication of research data shall always be based on FAIR principles. And then here metadata comes into play, right? Swedish Research Council recommendation on data management according to FAIR. So one can read through it, and I've also linked it here. But now everyone talks about FAIR, talks about everything. This has to be done this way. But what exactly happens when you do it in practice? So there are data management plans. You have to do documentation. You have to make it accessible. There has to be backup, etc., etc. But what are the issues that we face? Being fair does not mean being open, right? So a lot of questions come like, oh, does that mean I have to make my data open? Not necessarily, but you have to at least define it properly. So you should have levels of access, confidential, restricted, open. You should have appropriate licensing. If you don't want someone to just use your data, provide it proper licenses. Like define the degree of publicity you want, right? And then you always use identifiers. Now, what are the barriers? There are a lot of barriers which a lot of researchers fail. Can I give sharing rights? How? Form an agreement. Scooping. Can anyone take out, like, I give an idea, but can anyone take that idea and publish it as their own? No. Put it as the idea as like a digital object, or like write your idea, publish it somewhere so you have an object so you can cite it. Then I don't have time. What should I do? Sharing data saves time. Create a data management plan. I will come to it later, how you can save time. Then lack of incentives. What if I don't get any incentives? Even I share my data set, I don't have any incentive. No, but that also means if you have open data, you can also, now there are provisions to even cite data. And it has been there, for example, in genomics field and proteomics field, things like that, where you give a DOI of your data type in your papers, right? Then insecurity. Oh, what if I share my data and the person is not a trusted person? What will happen then? My data is too large. How will I share this? Split it into smaller chunks. Oh, my workflow is too complex. another person won't be able to understand. Give a proper README file. Oh, the whole process is unclear. Check with the library, check with data stewards, how many resources exist. There are different data types, for example. Then unclear value. Value is subjective, perspective are limitless. Opportunities are there for synthesis, for example, sensitive content and things like that, right? Now I move further. But what is the cost of not having fair research data? Almost 27 billion per year in Europe are wasted because the data is not reproducible. A person can't use the data. What does it mean? Some data is normalized, for example. So I don't have the proper data points. People give a graph, but don't list the raw data, for example. So these things affect especially people who take the experiments or data from others and then maybe use it for computational studies. Also, if you want to do a standardization, you need to have a data or data points which someone has done before. And then again, it becomes difficult, right? Now, there are two pillars of FAIR here. 20% are identifiers, which is digital objects, PIDs, codes, etc. But then 80% of this all comes down to metadata, right? So keeping this in mind, we move forward. Everyone knows, but just to give a brief recap, what is metadata? Simple terms, it is data about data. You define the data, right? From what the data was generated, how do samples differ, what and where the experiment was done, or what were the experimental conditions. So basically, it is minimum information that you provide to reproduce the data, sample information, protocols, etc. Now, there are different types of metadata. It could be like descriptive. What is the data set? Right? Structural. How is it organized? Then administrative. What are the rights? What is the licenses that is attached to it? Then provenance. How it was collected. And this is very critical for reuse. For example, if I say this particular experiment was done at 36 degrees, so to reproduce that, you have to say that this was the temperature it was done at. For example, how much dosage was given? Say, for example, 10 milligrams. So this is just an example of how the metadata looks like. Then there are different terms that describe your data. So a biologist would rather share a toothbrush with another biologist than share a gene name, which is very true. Like, I want to keep it so secret, right? But this is not the way to go forward. So what are different terms that you can use? Control vocabularies. So this is basically nothing but standardized and organized list of terms that you can describe to use data consistently, which improves data retrieval and sharing. Then there are the ontologies. Thesaurus simply means different or synonyms, for example, in simple terms. Then taxonomies like hierarchies, and these are very important for consistency and stringency, and also for making it machine actionable. And all these can be used a bit interchangeably. For example, ontology can also be a control vocabulary. So it captures relationships. For example, is a part of, contained by, produced by, and then hierarchy, right? But how does that mean? Like, it's really nice to say these things, but what does it mean in reality? So control vocabulary can have like a heading, basically in the middle. It could have different synonyms. Then there are different ontologies. And this is an example which I took from somewhere. And I thought this is really good for explanation. What an ontology means. For example, let's say there is a lung, right? But lung is a part of respiration organ, which is in fact a part of bigger or like organ system, which comes under anatomical structure. Okay, now I go further into classification, which is the lung part, like all the lung part. Is it pulmonary alveolus? Is it lung alveolus? And then different things like that. So you can have a whole ontology described here, and then maybe it is a part of, it is a subclass of. So this defines an ontology, to give an example, right? Okay, now there is how should you document your data? What is the information that you enter? So all this, talking about all this, what is a practical example? For example, you have a spreadsheet, right? So you have element and value of displays. What is the description of this? Which kind of data type it is? How much character length it is? What, in this part, what is the variable name? What is the sample ID? What is the patient ID? What is the sex of the patient? What is the date? when it was collected. So rather than just writing any date, like for example, some might write in the US standard way where month comes first. Some might write in Swedish standard way where the year comes first. But it is very good to define the format, right, which you will use. And if it is predefined in metadata, it's better for you. So you don't have clashes or there is no semantic interoperability issues, for example. So it's always good to use standards. Now, what is the research outputs and metadata in context? So how do you define these? Like I'm talking about it, but how in practice you can do it? So you start with the study and data design. Then you go to sample collection. Okay, so how should I collect the sample? This is an example of COVID-19, right? So you had like nasal swabs and things. Now, is it population samples? Is it one person each sample? Is it just like a smaller population sample, bigger population sample? What were the processing steps? How it was collected, right? Then how was the analysis done? What is the computational workflow? And everything has to be properly documented. And then the data goes into some repository, and preferably public repository. But having said all that, it is really, really important to keep in mind that everything should try to be made machine actionable, right? So if you have, say, for example, an RDF file, so then you can link all the open data, make it executable, make it discoverable, make it reproducible. How? Give everything digital objects attached. For example, sample ID. You can click on it, you go to the whole sample, for example. And there are different formats already available. There are different terminologies already available. You have guidelines for it in all different public repositories. What are examples of metadata? Sample information. Could it be organism? It could be tissue. It could be different conditions. What are the protocols? Now you know you can also share protocols, for example, and you can also have it as ELNs, in ELNs, right? We have protocols.io, for example, in SciLifeLab. Then what are the sequencing parameters? What are instrument models? What have you done any calibration? Different file formats, which are very important, and it's becomes an issue in the end when you are trying to submit the data. Do you require any ethical approvals, especially if it is sensitive data? What are the data processing steps and quality control logs? How do you do that? Everything said, simple terms, you have a sample, you do an experiment, later you do analysis, and then you do some submission, you find some submission tools, have proper metadata ready, do either compress the data files or the data files, raw data files, process data files, whatever, and then you submit into a repository. But having said that, where does metadata come in the whole research data lifecycle? Planning phase, determine what metadata is needed for doing the project and sharing data. But then collection, before even collecting, think about it. How will you have all of this documented, right? And then you will have to process and analyze it. Organize the metadata already in appropriate formats, using metadata to perform analysis. But all of this is telling you what to do, but what is it in actual terms? Means define standards and templates as soon as you're planning. Say, for example, you have genomic data, already start planning. Okay, this standard is somewhere there. I will come to it later also, but you should have all these standards in hand before you start. Creating metadata means capture the metadata at the source itself. Organizing metadata in appropriate formats mean you should document, you should have like scripts. If you have scripts, you should have it in a proper format. All the parameters, if you are creating, because it's a process, so maybe you have a lot of versions, and that's what happens in research. I did it. Something is wrong. Now I have to create version 2. Now I have to create version 3. You should already have that versioning, like version 1, version 2 available, right? Then using metadata to perform analysis means you already start, you do annotation, you do results, you should do all the plotting, then describing data sets, which is very important. You did everything, but if it's not described properly, it is not even reusable for yourself, right? So you should ensure that it's described properly. Also, the portability issues should not occur. deposit, and in the end, when you share, you should deposit data set with complete metadata attached. Now, having said all of these and giving an overview about what is metadata, how you should go about it, what are like, okay, I know all this, I've done all this, but what as an early researcher or a life science researcher, what should I start with? Practical steps. Start with small manageable steps. Don't think about the end goal, but start with very, very small steps. Then now you know that FAIR is such a big thing. So it's very important that as early as possible, you seek training in resources on FAIR principles, and then engage with research community to share best practices. So every research community is a bit different. For example, for some communities, there is already very good metadata practices. There is already very, very good standards which have been already established. For example, genomics, proteomics, but then there are some upcoming research fields, or because research is always evolving. And one problem I see in one field is, for example, imaging. Imaging doesn't have very proper standards. Where should I submit my raw data? Should I not submit the raw data, but just the processed files? So these are some upcoming research communities, right? So it's very important to engage with these research communities, not just to get the information, but also to give your own input there, right? So let's start with small manageable steps in data management. But first thing, having said that, don't think about other people. What will others do with my data? Think about yourself, right? Your own primary collaborator is yourself 6 months down the line. And your past self doesn't answer emails, right? I can ask from someone else, but not myself. So for me to understand this data and metadata after 6 months, after a year, think about 4 years down the line when I'm finishing the PhD. I don't even know what I did before 4 years, since I have not defined it properly. So not think about someone else, but think about yourself as the primary consumer. And what would I be able to understand it after 6 months? Would I be able to get everything after, say, 4 years? So this is something you have to keep in mind, your own future self, right? Now, having said that, let's go towards a web of FAIR and data and services. So make it machine actionable, as said. So this is like, does it work with my software? Some questions to answer. What type of research assets am I looking at? Where will people be looking for these? For example, if it's, say, for example, structural data, protein structures, biggest source is PDB. So people should be looking at PDB mostly, but you can also connect the data. For example, there is also one resource by Japan, right? Japanese PDB. Maybe some people will also look there. But let's see where mostly the data is shared, for example. Who should have access? This comes more in sensitive data kind of thing. But maybe you have still like semi sensitive kind of data. You don't want everyone to have access, or maybe it's pre-publication time. You still don't want everyone to have access. So who should have access? Which standards to use? This is very important. So you decide from beginning what standards to use. What cross references, what documentation, what README can you provide? Like, okay, everything is done. Do you have any protocols defined? Have you defined which steps to take? Where to look for? So basically, in hindsight, rich metadata. Now, discoveries are also made using shared data. So, like I said before, and I still emphasize on it, define a metadata standard early. Choose a standardized metadata schema relevant to your field, right? You can look at Dublin Core, you can look at DataCite, you can also look at discipline-specific metadata standards. I'll come to that later. Then, always use consistent naming conventions. Remember, like I said, it should be readable to your own future self, for example. So do you have versioning? Do you have a date, like provided proper dates? Oh, I did this on a certain day, but how do I remember that? Let's put a date when I like save a file. Document data from the start. Maintain a README file, and I would say to have like electronic lab notebooks. So you already document everything from the start. Automate wherever it is possible. And if someone doesn't know, like so some people are very much advanced in programming skills. Some people are not. So for people who are advanced, it comes to OpenRefine, Python scripts, R script, for example. But if you are not, then you can simply start with TSP files. You can simply start with simple text files, right? But you start somewhere. Then always try to store the data and metadata together, which is very important. So your data is somewhere, your metadata is somewhere, and then you won't be able to yourself figure out how to bring them together. Now, this requires data that is discoverable by both humans and machines. So always think that you can see it, but is it also machine actionable, which is also important. Then what are the different tools that you can manage for managing metadata? For example, there are spreadsheet templates. Like I said, electronic notebooks. Use as much as control vocabularies, as much as ontological terms as possible. Try to submit to already established repositories, which already provide you good metadata templates, good metadata structure. So you, and all, they have control vocabularies, they already have some ontology, so you can start there. Then also try to have automation tools like scripts, pipelines, so that you can submit data. And then always remember, data does not mean the data that you have done experiment on, but or you collected by experimentation, but also your protocols. If you are giving a presentation somewhere, slides, what algorithms you have developed, the document that is the README file, for example. So these all also count as data. And then they also deserve a good metadata. So getting data into desired state, what does that mean? You should always refine it. Like there should not be any formatting errors. There should not be some missing values. You should not have incorrect data in that. Is there any irrelevant data, right? So these things you should always check on. So it is not enough that you just submit the data, you have the metadata, but you should also check the quality of the data as well as metadata. So better metadata is for better data. Remember that. So there are a lot of community standards that are already there. You don't have to start from scratch. And then there are a lot of discipline-specific metadata standards. So this is just to give an idea, like almost there are 1,300 community standards, right? So I've listed some examples here. Then how do you decide how do you publish your data set in a data repository? Now I have my data set, which repository to choose? Okay. So this is kind of a small flow chart that we did together in our data stewards group. So how do you decide this? Okay, does my data contain any personal or sensitive information? Yes, go to restricted access repositories. Now we also have a federated EGA in NBIS, for example. So you can, if you have genomic data, you can submit it there. Then, okay, it's not. Is there any discipline-specific repository that exists? Yes. Which ones? ENA, PRIDE, Biosample, Protein Data Bank, like I talked about. So these could go there. So they already have standards. Okay, but my data does not fit any of these. What to do then? Then there are institutional data repositories. For example, we have SciLifeLab Figshare instance. So you can also submit there. Then SND has one. Even Stockholm University has. But no, it can't even go to institutional repository, or it does, my institute does not have one. Then there are general repositories now, like Zenodo, OSF, Figshare in general. So these, you can put your data there, right? Now let's do a small demo, like what you can do. I can quickly maybe share this in the chat. So this is just a small example I thought of. Let me share it in the chat.
I can do it,
Okay,
Parul.
thank you.
You can continue.
Okay, thanks.
Okay.
So it's just to give an example how you can decide, right? So it's just like a demo. You go here, answer these questions. So answer in the same as I have listed, and then you see which repository comes out. So if you don't know exactly which repository to look at, so you can also try to find it in this way. So maybe I give a minute or so. Okay, I think it should be fine. So this is just to give a small, because we all talk about things, but what about exactly a practical example? So this is a small practical example how you can decide on a repository, right? Now this is simply ENA. So ENA is a very good European repository, which is very much machine actionable, very well defined metadata, lot of checklists available. Checklists mean that you have different metadata standards for different type of genomic data. So genomic data doesn't mean one data type. It has a lot of things like wastewater, COVID-19, uh, say for example, uh, a disease, for example, which is attached. So this is a lot of machine actionability that is there. So what is machine actionable here? This is XML format, right? And then there is XML study. So you also have cross-referencing here. Then you can also connect your own ORCID to it. Are there any related DNA records? Yes. So these all come under here. And when you have all, why do you have this show and show here? So you at least you can connect it. So you can go to different repositories, right? So this makes it all very machine actionable. Okay. So this is just an example of a fair resource. Now, talking about all this, how can it connect back to you? I talked about metadata, you can select some repository, you can do everything, but how does it connect to the whole network when you have good metadata? So once you have good metadata, you can define the relationships or connection between entities, right? So data set is cited by some paper, which has a DOI. So this paper is published by some organization that has a ROR ID, right? Also, your data set that you have produced was funded by some funder, which also is an organization, which also has a ROR ID. Okay, now how does this all connect back to you as a person? Have a ORCID ID. So you can connect this data set to your ORCID ID, which I just showed in ENA, right? So you had an ORCID ID there. So you already have everything, all your research outputs listed in ORCID ID. Again, you are as a person affiliated to organization. Say, for example, not example, but I am affiliated to SciLifeLab. SciLifeLab has a ROR ID. So now I have everything, all the connections made in one place. And this basically, I don't have to look in different parts or different places to get all my data, but I have connected everything to ORCID, for example, right? So this is so important to have your own research outputs in one place. So how a paper, again, I lost my, oh, this is really, okay. So a paper cites a data set, a person authors a paper, a person is affiliated with an institution funds a research output, but how all this connect is a simple diagram here. So that now you see, like with your own context, how much metadata is important for your own research outputs, right? Okay, now let's come to second part. Everything said and done. Some people might be on different levels, so you should seek training and resources on FAIR principles, because for you, FAIR principle, as much as you do training, as much as you can understand the importance, you can understand how to implement all these. Like talking about it, thinking about it is not enough, but how should I be able to apply this in practical area? Then you have to understand FAIR. You have to understand to use the metadata repositories, like even institutional, Figshare, Dryad, Dataverse, for example, also like discipline specific. You can adopt different standards. For example, for DOIs, there is ORCID ID, like I said, there is ROR ID, there is, for example, rates. Then you should always check licensing and permission. For example, if a lot of presentations are shared in SciLifeLab instance of Figshare, so they mostly have CC BY, but maybe some have other licensing. So you should always be aware of which licensing to use. Then it is very good to always attend training workshops. So many universities provide that. Also, we have some from Training Hub, which is Introduction to Data Management Practice, Open Science in Swedish context, for example. There are too many resources out there now, like FAIR has become a de facto global norm. There is RDA, Research Data Alliance, which is working a lot towards different metadata standards. There is a lot of research communities here. People go in it. There are data steward communities to define as much as proper metadata as possible. Go FAIR, which gave these FAIR principles, then FAIR in practice, for example. There are different reports published, how it is important. So one can go through these, right? Now, another thing, if you are still not sure, you can engage with research communities to share best practices, right? What does this mean? The vision is here to promote semantic technologies to achieve fair and engaged stakeholders. Purpose here to address shared challenges, promote FAIR principles, and support metadata solutions. So goal is to build expertise, foster collaborations. This is more about SciLifeLab Data Community. And I've also given a QR code here if someone wants to join and work together to develop good metadata standard. Not even metadata standard, but to maybe improve on one, because we don't want to start from scratch. Then we could have like communication channels, we could have outreach programs. Then more also, it could be personalized support. We already have, for example, NBIS drop-ins, but we could also, if someone writes, could also have personalized support, could have a point of contact to connect. And then what is the state of FAIR, for example? So this is something, if you want to contribute, you can join and also learn and also make contribution here. Then what are the challenges which are addressed for FAIR metadata? Because having said everything, still there are a lot of challenges to deal with, otherwise we won't need it, right? There are inconsistent standards, there is incomplete metadata. Then there is very much limited machine actionable formats. Also, there are people who are not so maybe capable of making it machine actionable so much because of the fact there are not many machine actionable formats. Then there could be interoperability issues, right? Then there are not enough vocabularies. What is the state of FAIR metadata right now? So we need to have progress in standard and tools, but to build on the already existing ones. Then have it as much as automated as possible. So you just go in the drop down, you select what is more relevant to you, and you do not have to do manual input as much as possible. So that makes it as much as machine actionable as one could, right? So we have in this, we already have some connections with different communities outside SciLifeLab, like LeafFace, GoFair, GA4GH, which had a conference recently. We are already engaged with RDA EOSC. We are also involved with EMBL and VIB, which is from Belgium. EOSC, again, then there is SND and RAID. SND is Swedish National Data Network. Then we also have like different ones here. Okay, and why and how to connect? Okay, I have already listed a lot of things why you should connect. Like you should be able to yourself provide some input, but also gather back from these communities. Then you can also join different forums, share resources through workshops and things like that. Having said all of this, having this big presentation, what is a small summary? Like I always like to think about, okay, I have listened to everything, but what should I take back from this? Okay. Think about FAIR principles. FAIR principles make your research future-proof. Start simple, be consistent, use standards. Don't think about the end goal right now. Only just start somewhere. Metadata is not extra work. It's part of a good research practice. Like I said, your own future self will thank you. Don't do it for someone else. Do it for yourself, your own future. Down 6 months, 1 year, 4 years down the line. Then small improvements, like small things like putting a date or having version, right? These small improvements will save you a huge time and frustration later, right? So this is from me, and thank you. And you can also have, if you have any questions, you can share.